Basics of Medical Biochemistry. First edition, 2023. Chapter 12. Metabolism of Lipids. Part 1 of 3. By Professor Dr. Muhammad Jawad. MBBS, Tao, M.Phil Philosophy, Ph.D, Dip M.E. Head of the Biochemistry Department-Fazaia Ruth Pfau Medical College Air University, PAF Base Faisal-Karachi, Pakistan. Ex-Professor of Biochemistry, Qassim University, Kingdom of Saudi Arabia. Ex-Professor, Head of the Biochemistry Department and Vice Principal. Liaquat College of Medicine and Dentistry and Fatima Jinnah Dental College, Karachi, Pakistan. Chapter 12. Metabolism of Lipids. 12.1 Introduction. Lipids are a heterogeneous group of compounds, hydrophobic in nature, i.e., insoluble in water but soluble in nonpolar solvents like acetone, alcohol, benzene, chloroform, etc. Lipids yield more than twice as much energy for the same mass as carbohydrates or proteins. Sources of lipids. Main lipids, such as triglycerides or triacylglycerols, TAG, phospholipids, and cholesterol are obtained from the diet. Dietary sources of lipids include butter, cream, cheese, oil, animal fats, etc. The main function of dietary lipids is to provide essential fatty acids and to improve the absorption of fat-soluble vitamins. Cholesterol is quite essential for life, however, its excess in the body is associated with many pathologies, particularly cardiovascular disease. De novo synthesis of cholesterol takes place in the liver from acetyl-CoA. Cholesterol acts as a precursor for bile salts, steroid hormones, and vitamin D. Under normal circumstances, a complex balance is maintained between the biosynthesis, utilization, and transport of cholesterol and other lipids to avoid harmful excess. Storage of lipids. A significant amount of lipids is stored in adipose tissue. Adipocytes are fat-storing cells having lipid droplets composed of a central core of triacylglycerol and cholesterol esters surrounded by a layer of phospholipids and a protein coat called paralipins. Fat depots are mainly found underneath the skin, in the omentum, around the heart and kidneys. Adipose tissue also serves as protection against heat loss and mechanical shock. Lipids in plasma. In the plasma, lipid exists in two forms: free fatty acids, FFAs, and lipoproteins. Types of lipoproteins are chylomicron, very low-density lipoprotein, VLDL, low-density lipoprotein, LDL, and high-density lipoprotein, HDL. Transport of lipids. Free fatty acids are not free in real, they are bound with albumin. Lipoproteins are the moving fuel in the body. Chylomicrons are found in the blood as well as in lymphatic fluid, where they serve to transport fat from its port of entry in the intestine to the liver and to adipose tissue. HDL is involved in reverse cholesterol transport, i.e., takes up cholesterol from peripheral tissues and returns it to the liver, thus HDL is termed as good cholesterol. 12.2 Lipolysis of depot fat. Lipolysis is the catabolic aspect of lipid metabolism and describes the process by which fats are broken down by enzymes and water, hydrolysis. Lipolysis occurs in adipose tissue, which are the fatty tissues that cushion and surrounds different organs. It may also occur in muscle or liver, where a smaller amount of fat is stored. Lipolysis causes the release of fatty acids and glycerol from the breakdown of triglycerides stored in adipose tissue. Triglycerides are transported through the blood to appropriate tissues, adipose tissue and muscle mainly, by lipoproteins such as VLDL. Mobilization of fats from fat deposits takes place under the influence of two hormones, glucagon and adrenaline. These hormones act through the activation of adenylate cyclase, which results in the formation of CAMP. These hormones bind to a GS-coupled receptor, resulting in increased CAMP and increased protein kinase activity. The protein kinase brings about the phosphorylation of paralipin, which results in the movement of hormone-sensitive lipase, HSL, up to the surface of the lipid droplet. The HSL is found in adipose tissue. It is activated by epinephrine, norepinephrine, cortisol, growth hormone, and thyroid hormone. Triglycerides present in the VLDL undergo lipolysis by the cellular lipases, i.e., HSL of target tissues, which yields glycerol and free fatty acids. Site of occurrence and importance of lipolysis. The major site of lipolysis is adipose tissue. Lipolysis increases in response to starvation. Adult man ingests about 60 to 150 g of lipids per day, of which 90% is normally TAG. The remainder of dietary lipids are made of cholesterol, cholesterol esters, phospholipids, and FFAs. Lipids are energy-rich nutrients having a caloric value of 9.3 kcal/g, which is almost twice carbohydrates and proteins. Triacylglycerols constitute about 10 to 12 kilograms of total body weight. The energy available from stored fats is about 85% of the total energy available in the body. In mammals, the major site of accumulation of triacylglycerols is the cytoplasm of adipose cells, fat cells. Droplets of triacylglycerol coalesce to form a large globule, which may occupy most of the cell volume. Adipose cells are specialized for the synthesis and storage of triacylglycerol and for their mobilization into fuel molecules that are transported to other tissues by the blood. 2.2.1 Steps of lipolysis. 3 different lipases hydrolyze the triacylglycerol to 3 fatty acids and glycerol. Lipolysis in adipocytes is the first reaction catalyzed by the hormone-sensitive lipase. The diacyl and monoacylglycerol lipases are present in excess, so, once the hormone-sensitive lipase is activated, lipolysis of the triacylglycerols goes to completion. 2. Intestinal lipase acts on 1 monoacylglycerols, converting them into glycerol and free fatty acids. Thus, the end products of triacylglycerols are A2 monoacylglycerols, 72%. B1 monoacylglycerols, 6%. C glycerol and free fatty acids, 22%. 3 phospholipase A1, A2, C and D, they hydrolyze various phospholipids. Figure 12.1 The process of lipolysis. 2.2.2 Product of lipolysis and their fate. The product of lipolysis is the free fatty acids and free glycerol. Both products leave the adipocyte. Most tissues use fatty acids mainly as a metabolic fuel for energy production, except the brain due to the blood-brain barrier. The glycerol is taken up by the liver or kidney and used as a substrate for gluconeogenesis or reesterification. Normal fasting plasma free fatty acid level, 15-20 mg/dl, is increased by anti-insulin hormones, epinephrine, glucagon, cortisol, growth hormone, thyroxine, starvation and diabetes mellitus. The plasma-free fatty acid level is decreased by insulin. Fatty acids are also used for protein modification. The esterification of fatty acids in adipose tissue to form TAGs is dependent on active carbohydrate metabolism for the formation of dihydroxyacetone phosphate as the only source of glycerol 3-phosphate for the synthesis of TAGs. The entry of glucose into the adipose tissue cells needs a glucose transporter, GLUT4, insulin-dependent, is an essential requirement for TAG synthesis in adipose tissue. The TAG stores in adipose tissue are continually undergoing lipolysis, hydrolysis, and reesterification. Thus, there is a continuous cycle of lipolysis and esterification within the tissue. The metabolism of fatty acids, therefore, consists of catabolic processes which generate energy and primary metabolites from fatty acids, and anabolic processes which create biologically important molecules from fatty acids and other dietary carbon sources. 12.3 Glycerol metabolism. The glycerol absorbed from the diet or derived from lipolysis of fat enters the bloodstream and is absorbed by the liver or kidney, where it is converted to glycerol 3-phosphate by the enzyme glycerol kinase. The glycerol cannot be used by adipose tissue cells as there is no glycerol kinase. Hepatic glycerol 3-phosphate is converted mostly into dihydroxyacetone phosphate, DHAP. This reaction is catalyzed either by a cytoplasmic glycerol phosphate dehydrogenase, or a similar enzyme present in the mitochondria. In addition to their different locations, these two dehydrogenase enzymes differ in that a different coenzyme accepts the electrons removed from glycerol phosphate. In the case of cytoplasmic dehydrogenase, NAD+ accepts the electrons and is reduced to NADH plus H+, whereas, in the case of mitochondrial dehydrogenase, flavine adenine dinucleotide, FAD, accepts the electrons which are reduced to FADH2. Triose phosphate isomerase converts dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. Either glyceraldehyde 3-phosphate or dihydroxyacetone phosphate may be used as substrates for glycolysis or gluconeogenesis. Thus, the glycerol part of fat is metabolized through the metabolic pathway of carbohydrates. Figure 12.2 Metabolism of glycerol, NAD, nicotinamide adenine dinucleotide phosphate, ADP, and ADP, adenosine tri- and diphosphate, P, phosphate. 12.4 Oxidation of fatty acids. Fatty acids are oxidized by many methods like oxidation, and omega oxidation. The process of oxidation is different for even carbon, odd carbon, saturated and unsaturated fatty acids. Most of the fatty acids are catabolized by oxidation in mitochondria. However, some fatty acids are oxidized in peroxisomes and endoplasmic reticulum. 12.4.1 Oxidation of even carbon-saturated fatty acids in mitochondria. The oxidation of even carbon-saturated fatty acid can be described in the following three phases. I. Activation of the fatty acid. 2. Transport of fatty acyl CoA to the mitochondrial matrix. 3. Repeated oxidation of acyl CoA, in spiral fashion, to produce acetyl CoA, FADH2 and NADH. I. Activation of fatty acid. Activation of fatty acid takes place in the cytosol. Fatty acid reacts with coenzyme A to produce high-energy fatty acyl CoA. The enzyme is fatty acyl CoA synthetase. 1 mole of ADP and 2 high-energy phosphate bonds are utilized in activation. Figure 12.3 Activation of fatty acid, AMP, adenosine monophosphate, PPI, pyrophosphate. The difference between the designation acyl CoA and acetyl CoA is that acyl refers to a random-length fatty acid carbon chain that is covalently bonded to coenzyme A, whereas acetyl refers to a 2-carbon chain covalently bonded to coenzyme A. Figure 12.4 Difference between acyl CoA and acetyl CoA. 2. Transport of fatty acyl CoA to the mitochondrial matrix. Fatty acyl CoA is too large to pass through the inner mitochondrial membrane to the mitochondrial matrix, where the enzymes required for fatty acid oxidation are located. This problem is looked after by a transporter called carnitine. Role of carnitine in the transport of fatty acyl CoA through the inner mitochondrial membrane. Carnitine, beta-hydroxygamma-trimethylammonium butyrate, CH3, 3N plus CH2CH, O, CH2Cu, is widely distributed in nature and is particularly abundant in muscle. It is mainly obtained from animal-based foods. It is also synthesized endogenously. Fatty acyl CoA combines with carnitine to form fatty acyl carnitine with the release of coenzyme. This reaction takes place in the intermembrane space of mitochondria by the enzyme carnitine acyltransferase I. The fatty acyl carnitine is translocated into the mitochondrial matrix as carnitine moves out. Carnitine acyltransferase II on the inner mitochondrial membrane transfers the fatty acyl group back to coenzyme A, to form fatty acyl CoA in the matrix. Inhibitor of carnitine transport. Malonyl CoA is the inhibitor of the enzyme carnitine acyltransferase I. Malonyl CoA is the substrate for fatty acid synthesis. Thus, at the time of fatty acid synthesis, fatty acid oxidation is decreased. Figure 12.5 Role of carnitine in the transport of fatty acyl CoA through the inner mitochondrial membrane, PPI, pyrophosphate. 3. Oxidation of fatty acyl CoA. Reactions of the beta-oxidation pathway. The successive oxidation of fatty acyl CoA takes place through the following steps. Step 1. Oxidation of fatty acyl CoA, dehydrogenation. In this reaction, H atoms are removed from the N carbons. A trans C=C bond is formed between N carbon atoms of the product known as transenvoyl CoA. The enzyme is dehydrogenase and FAD is reduced to FADH2. Figure 12.6 Oxidation of fatty acyl CoA to transenvoyl CoA, FAD. flavine adenine dinucleotide. Step 2. Hydration of trans-in-oil CoA. The addition of water takes place across the trans double bond by the enzyme hydratase, producing a secondary alcohol at the carbon position. As a result, hydroxyacyl CoA is produced. Figure 12.7 Hydration of trans-in-oil CoA to hydroxyacyl CoA. Step 3. Second oxidation, dehydrogenation. In this step, the hydroxyl group is oxidized to a keto functional group to produce ketoacyl CoA. The enzyme for this reaction is dehydrogenase and the hydrogen acceptor is NAD+, which is changed into NAD plus H+. Figure 12.8 Oxidation of hydroxyacyl CoA to ketoacyl CoA. Step 4. Thiolysis, chain cleavage. The cleavage of ketoacyl CoA occurs by splitting the bond between the alpha and beta carbons. This yields acetyl CoA and a fatty acyl CoA shorter by 2 carbons. The enzyme involved is thiolase. Figure 12.9 Cleavage of ketoacyl CoA to acetyl CoA and a new fatty acyl CoA shorter by 2 carbons. The acetyl CoA produced enters the TCA cycle and the new molecule of fatty acyl CoA, shorter by 2C, repeats the same sequence again, each time losing 2 carbon atoms as acetyl CoA until the entire molecule is completely oxidized. Bioenergetics of stearic acid, 18C saturated fatty acid. In oxidation, 8FADH2 equals 8 times 1.5 equals 12 ADP, by etc. 8NADH equals 8 times 2.5 equals 20 ADP, by etc. It consumes 2 ADP to activate the free stearic acid, ADP rightward arrow AMP plus PPI. The net yield of energy is 32 equals 30 ADP per mole of stearic acid. In TCA cycle, 9 acetyl CoA molecules, from beta oxidation, are oxidized by TCA to give 9 times 10 equals 90 ADP. Total moles of ADP produced from the complete oxidation of 1 mole of stearic acid equals 30 plus 90 equals 120. Figure 12.10 Beta oxidation of stearic acid, 18C, showing the production of 9 acetyl CoA, 8FADH2 and 8NADH plus H+, source, slideplayer.com. 12.4.2 Oxidation of odd carbon saturated fatty acids in mitochondria. Most of the fatty acids oxidized in humans are even carbon saturated fatty acids. In the case of fatty acids containing an odd number of carbon atoms, they follow the same steps of oxidation except that the final products are acetyl CoA and a 3-carbon fragment as propionyl CoA. The propionyl CoA is changed in a series of reactions to succinyl CoA, which enters the TCA cycle, as does the acetyl CoA. Following are the 3 reactions by which propionyl CoA is oxidized. Propionyl CoA is carboxylated by propionyl CoA carboxylase to form the D-stereoisomer of methylmalonyl CoA. Biodin acts as the cofactor for carboxylase. There is the formation of the carboxybiodin intermediate that needs energy, which is provided by ADP. Dimethylmalonyl CoA is changed into L-methylmalonyl CoA by the enzyme methylmalonyl CoA epimerase. L-methylmalonyl CoA undergoes an intramolecular rearrangement to form succinyl CoA, which enters the citric acid cycle. This rearrangement is catalyzed by methylmalonyl CoA mutase, which requires vitamin B12 as coenzyme. The propionyl CoA is also involved in the synthesis of porphyrins and the utilization of acetoacetate. Figure 12.11 Beta oxidation of odd carbon saturated fatty acid, 3C propionate. 12.4.3 Oxidation of monounsaturated fatty acids in mitochondria. The presence of a double bond creates a problem, which is overcome by epimerase and isoenzymes. Oxidation of monounsaturated fatty acyl CoA requires additional reaction performed with the help of the enzyme isomerase. Double bonds in the unsaturated fatty acids are in the cis configuration and cannot be acted upon by in-oil CoA hydratase, the enzyme catalyzing the addition of water to the trans double bond generated during beta oxidation. In-oil CoA isomerase repositions the double bond by converting the cis isomer to trans isomer, a normal intermediate in beta oxidation. Energy production by unsaturated fatty acid is less than by saturated fatty acid. 12.4.4 Oxidation of polyunsaturated fatty acids in mitochondria. The oxidation of polyunsaturated fatty acid requires two additional reactions and another enzyme reductase, along with isomerase. Consider the example of beta oxidation of linoleic acid, an 18C polyunsaturated fatty acid with cis-delta 9 and cis-delta 1,2 double bonds. Its oxidation involves the following steps. The cis-delta 3 and cis-delta 6 double bonds, old positions were on C9 and C12, appeared after 3 cycles of beta oxidation in 12C fatty acyl CoA. 2. The cis-delta 3 is converted into a trans-delta 2 double bond by isomerase, cis-delta 6 remains same. 3. Another round of beta oxidation produces 10C fatty acyl CoA with double bonds as cis-delta 4 and trans-delta 2. 4. The NADF-dependent 2,4-dienoyl CoA reductase adds 2 H atoms to remove cis-delta 4 and at the same time trans-delta 2 becomes trans-delta 3. V isomerase shifts trans-delta 3 to trans-delta 2. Now the remaining structure is oxidized on the usual pattern of beta oxidation. Figure 12.12 Beta oxidation of linoleic acid, 18C polyunsaturated fatty acid. 12.4.5 Oxidation of fatty acids in peroxisomes. Peroxisomes are responsible for the oxidation of very long-chain and branched-chain fatty acids. The oxidation of fatty acids in peroxisomes is the same as in mitochondria, with the following distinctive features. FADH2 produced in beta oxidation does not enter the electron transport chain, etc., but rather combines with O2 directly to form H2O2. Peroxisomes do not have etc. like mitochondria. 2H2O2 dissociates into water and oxygen by catalase. No ADP is produced, but energy is liberated as heat in this step. 2H2O2 2H2O plus O2. 3 NADH plus H plus and acetyl CoA generated in peroxisomes are shifted to the cytosol and then to mitochondria for oxidation and energy production. 4. Oxidation in peroxisomes is induced by a high-fat diet and anti-hyperlipidemic drugs, example clofibrate. Peroxisomes can also metabolize decarboxylic fatty acids, prostaglandins, leukotrienes, and the carboxyl side chains of certain xenobiotic and the bile acid intermediates D and trihydroxycoprostanic acids. 12.4.6 Alpha oxidation of branched-chain fatty acids in peroxisomes. Alpha oxidation is important in the catabolism of branched-chain fatty acids such as phytanic acid, which is a 20C branched-chain fatty acid derived from the dietary sources. Its catabolism through alpha oxidation involves the following steps. Phytanic acid is activated to phytanoyl CoA by phytanoyl CoA synthetase. 2. The phytanoyl CoA is converted to 2-hydroxyphytanoyl CoA by the enzyme phytanoyl CoA 2-hydroxylase in a reaction that requires 2-oxyglutarate and oxygen. Succinate and CO2 are released. 3. 2-hydroxyphytanoyl CoA is degraded by the enzyme 2-hydroxyphytanoyl CoA lyase to pristanyl. Formyl CoA is also released. 4. Pristanyl is oxidized to pristanoic acid by pristanyl, aldehyde, dehydrogenase. This enzyme needs NAD+ or NADP as a coenzyme. Further oxidation of pristanoic acid in peroxisome takes place on the usual pattern of fatty acid beta oxidation. Figure 12.13 The alpha oxidation of phytanic acid, 20C branched-chain fatty acid, in the peroxisome. The biological significance of alpha oxidation. During alpha oxidation of fatty acids, several hydroxy fatty acids are produced as intermediates of this pathway such as cerebronic acid. Cerebronic acid can be used as a precursor for the synthesis of cerebrosides and sulfatides. Similarly, odd-chain fatty acids produced upon decarboxylation during this pathway can be used for the synthesis of sphingolipids and can also undergo beta oxidation to form propionyl CoA and acetyl CoA. These products are metabolized through the TCA cycle. 12.4.7 Omega oxidation of fatty acids in the endoplasmic reticulum. this type of oxidation occurs at the methyl end of the fatty acid molecule, in the endoplasmic reticulum of liver and kidney cells. By the action of various cytochrome P450 isozymes in the endoplasmic reticulum, the terminal methyl group of a fatty acid chain is oxidized to a carboxylic acid. The end product is decarboxylic acids, which are shifted to peroxisomes, where they are degraded by beta oxidation from the omega end. Omega oxidation is quantitatively less significant in humans. This pathway of fatty acid oxidation mainly occurs in bacteria and fungi. Figure 12.14 Omega, oxidation of fatty acid in the endoplasmic reticulum. The biological significance of omega oxidation: it represents a minor pathway of overall fatty acid oxidation. But, in certain clinical disorders, such as diabetes, chronic alcohol consumption and starvation, this pathway becomes an effective route for the removal of toxic levels of free fatty acids. Clinical aspects: 1. Deficiency of carnitine or carnitine transferase or translocase. Carnitine deficiency is seen in newborn and preterm babies due to inadequate synthesis or renal loss. Hypoglycemia and hypoketosis are common findings. Symptoms include muscle cramps during exercise, severe weakness, and death. Muscle weakness is related to the importance of fatty acids as a long-term energy source. Management includes oral supplementation with carnitine and recommendation of a diet containing medium-chain fatty acids since they do not require a carnitine shuttle to enter mitochondria. 2. Jamaican vomiting sickness. It is caused by dietary consumption of the unripe fruit of the ackee tree, which contains the toxin hypoglycin, which inactivates medium and short-chain acyl-CoA dehydrogenases, inhibiting beta-oxidation that results in hypoglycemia. 3. Decarboxylic aciduria. It is characterized by the excretion of C6-C10 decarboxylic acids and non-ketotic hypoglycemia, which is caused by a lack of mitochondrial medium-chain acyl-CoA dehydrogenases. 4. Refsum's disease. It is a neurocutaneous syndrome that is characterized biochemically by the accumulation of phytanic acid in plasma and tissues. This disorder is due to the deficiency of phytanoyl CoA2 hydroxylase, and patients are unable to degrade phytanic acid. The main clinical features are peripheral polyneuropathy, cerebellar ataxia, retinitis pigmentosa, and ichthyosis, rough, dry, and scaly skin. The symptoms develop gradually and insidiously from childhood through adolescence and extend up to early adulthood. Considerable improvement can be achieved when patients are kept on low phytanic acid diets for prolonged periods, example diets that exclude dairy and ruminant fat. 5. Zellweger syndrome. There is an inherited absence of peroxisomes in all tissues of the body. Long-chain fatty acids are not oxidized. Symptoms occur due to the accumulation of long-chain fatty acids in brain tissue. 12.5 Biosynthesis of fatty acids. Synthesis of fatty acids takes in the cytosol of many organs, including the liver, kidney, brain, lung, mammary gland, and adipose tissue. Acetyl-CoA is the immediate substrate, and it is mainly derived from carbohydrates and proteins when these are ingested more than the caloric requirement. Acetyl-CoA obtained from beta-oxidation of fatty acids is not considerably involved in the synthesis of fatty acids. The body can synthesize all fatty acids except essential fatty acids. Acetyl-CoA is produced in mitochondria. Figure 12.15 Formation of acetyl-CoA as a source of fatty acids synthesis. Requirements for fatty acids synthesis. These include acetyl-CoA as a precursor, the main enzyme fatty acid synthetase complex. Another enzyme needed is acetyl-CoA carboxylase. Several cofactors such as NADP+, H+, ADP, Mn2+, Mg2+, biotin, HCO3, as a source of CO2, and carrier protein known as acyl carrier protein, ACP. Site of formation of acetyl-CoA. Acetyl-CoA is produced from pyruvate, ketogenic amino acids, fatty acid oxidation and by alcohol metabolism in the mitochondria. In order to be utilized for the synthesis of fatty acid, it must come to the cytosol. Transport of acetyl-CoA from mitochondria to cytosol. The inner membrane mitochondrial is impermeable to acetyl-CoA. The entry of acetyl-CoA into the TCA cycle depends on the availability of oxalostate. The concentration of oxalostate is lowered if the carbohydrate is unavailable, starvation, or improperly utilized, as in diabetes. Oxalostate is normally formed from pyruvate by pyruvate carboxylase, an anaplerotic reaction. It means that fats burn in the flame of carbohydrates. Inside the mitochondria, acetyl-CoA first reacts with oxalostate to form citrate, as a part of the reaction of the TCA cycle catalyzed by citrate synthase. Citrate is transported to the cytosol by crossing the inner mitochondrial membrane with the help of a citrate transporter. In the cytosol, citrate is cleaved by citrate lyase to give acetyl-CoA and oxalostate. The oxalostate is involved in one of the following metabolic pathways. Utilized in glucose production after being converted to phosphenol pyruvate and the other glycolytic intermediates. Reduced to malate by malate dehydrogenase. Involved in the synthesis of non-essential amino acids. It provides the point of entry for aspartate and asparagine to the TCA cycle. It acts as the precursor of purines and pyrimidines via aspartate. Figure 12.16 Transport of acetyl-CoA from mitochondria to cytosol. Sources of NADPH plus H+, HMP shunt is the major source of NADPH plus H+. It is actively occurring in organs involved in lipogenesis such as the liver, adipose tissue, lactating mammary gland, etc. Conversion of malate to pyruvate catalyzed by the malic enzyme, NADP malate dehydrogenase, and the extramitochondrial isocitrate dehydrogenase reaction is also the sources of NADPH plus H+. Figure 12.17 Sources of NADPH for fatty acid synthesis. Fatty acid synthetase complex, it is the main enzyme of fatty acid synthesis. The human fatty acid synthetase complex is a homodimer cytosolic enzyme that regulates the de novo biosynthesis of long-chain fatty acids. It is composed of two identical monomers, polypeptide chains, and each one is equipped with 7 enzyme activities of fatty acid synthetase. Synthesis of all enzymes in the complex is well coordinated as it is encoded by a single gene. Following are the names of 7 enzymes of fatty acid synthetase complex. I-acetyl-CoA-ACP transacylase, 2-malonyl-CoA-ACP transferase, 3-beta-ketoacyl-ACP synthase, 4-beta-ketoacyl-ACP reductase, V-beta-hydroxybutyryl-ACP dehydratase, 6-envoyl-ACP reductase, 7-thioesterase or deacylase. 1.2.5.1 Biosynthesis of saturated fatty acids. The synthesis of palmitic acid, which is a 16C saturated fatty acid, occurs through the following steps. 1. Formation of malonyl-CoA. The acetyl-CoA is carboxylated to malonyl-CoA by the enzyme acetyl-CoA carboxylase. This reaction needs ADP and occurs in the presence of biotin and Mn2+ ions. Acetyl-CoA plus CO2 plus ADP malonyl-CoA plus ADP plus pi. Figure 12.18 Formation of malonyl-CoA. The nature of the reaction is condensation and it occurs in 2 steps, carboxylation of biotin involving ADP and then the transfer of the carboxyl to acetyl-CoA to form malonyl-CoA. 2. Formation of acetyl-ACP. It occurs by acetyl-CoA-ACP transacylase as shown in the following reaction. Acetyl-CoA plus ACP acetyl-ACP plus CoA. 3. Formation of malonyl-ACP. It is formed by the condensation of malonyl-CoA with ACP catalyzed by malonyl-CoA-ACP transferase. Malonyl-CoA plus ACP malonyl-ACP plus CoA. The next 4 steps will be condensation, reduction, dehydration, and reduction. These steps are repeated till a 16C fatty acid is synthesized. Figure 12.19 Formation of malonyl-ACP and acetyl-ACP, ACP, acyl carrier protein. 4. Formation of acetoacetyl-ACP, it is formed by the condensation of acetyl-ACP with malonyl-ACP. This reaction is catalyzed by beta-ketoacyl-ACP synthase. Acetyl-ACP plus malonyl-ACP acetoacetyl-ACP plus ACP plus CO2. Decarboxylation in this step allows the reaction to go to completion, pulling the whole sequence of reactions in the forward direction. Acetoacetyl CoA is a 4C fragment. 5. Formation of beta-hydroxybutyryl ACP. The reduction of acetoacetyl ACP produces beta-hydroxybutyryl ACP. The reaction is catalyzed by beta-ketoacyl ACP reductase. The coenzyme for this reaction is NADPH plus H+. Acetoacetyl ACP plus NADPH plus H plus beta-hydroxybutyryl ACP plus NADP plus. 6. Formation of trans-2-butanoyl ACP. The dehydration of beta-hydroxybutyryl ACP by the enzyme beta-hydroxybutyryl ACP dehydratase results in the formation of trans-2-butanoyl ACP. The process of dehydration causes the creation of double bonds in the product. Beta-hydroxybutyryl ACP trans-2-butanoyl ACP plus H2O. 7. Formation of butyryl ACP. The reduction of the double bond of trans-2-butanoyl ACP takes place in this reaction to form butyryl ACP. The reaction is catalyzed by anoyl ACP reductase. It is another NADPH plus H plus dependent reaction. Trans-2-butanoyl ACP plus NADPH plus H plus butyryl ACP plus NADP plus. Figure 12.20 Formation of butyryl ACP. 8. Chain elongation of butyryl ACP to 16C palmitic acid. Now the second round of fatty acid synthesis begins with the repetition of steps 4 to 7. Another molecule of malonyl ACP is added to butyryl ACP to elongate it to a 6C fatty acid derivative along with the loss of 1 CO2. The 3-ketoacyl group is reduced, dehydrated, and reduced again, steps 5, 6, 7, to form the 6C fatty acyl ACP. The above 4 steps of chain elongation are repeated so that a 16C fatty acyl, palmitoyl, ACP is formed. It is separated from the enzyme complex by the activity of a 7th enzyme of the fatty acid synthetase complex, i.e., thioesterase or deacylase. Under normal circumstances, chain elongation is stopped at this point, and free palmitate is released from the ACP molecule. To a lesser extent, further elongation of palmitic acid to 18C stearic acid takes place actively in the endoplasmic reticulum, but it may also occur in the cytosol as well as in mitochondria. The source of 2 carbons for this elongation is malonyl CoA. The net reaction of palmitic acid synthesis: 8 acetyl CoA plus 7 ADP plus 14 NADPH plus H plus palmitic acid plus 8 CoA plus 14 NADP plus 7 H2O. Plus 7 ADP plus 7 pi. 12.5.2 Regulation of fatty acid synthesis. The rate-limiting step in the biosynthesis of fatty acids is the reaction catalyzed by acetyl CoA carboxylase. Allosteric control. Citrate is an activator while palmitoyl CoA acts as a feedback inhibitor of the acetyl CoA carboxylase. 2. Phosphorylation. Acetyl CoA carboxylase is inactivated by phosphorylation carried out by adenosine monophosphate activated protein kinase, AMPK. In the presence of hormones such as glucagon and epinephrine, acetyl CoA carboxylase is phosphorylated and becomes inactive. Insulin stimulates fatty acid synthesis by stimulating protein phosphatase that activates acetyl CoA carboxylase via dephosphorylation. 3. High carbohydrate and low-fat diet. Synthesis of acetyl CoA carboxylase and then fatty acids production increased. 4. Fasting and high-fat diet. Synthesis of acetyl CoA carboxylase is decreased. 12.5.3 Biosynthesis of unsaturated fatty acids. The synthesis of unsaturated fatty acids takes place by the process of unsaturation of corresponding saturated fatty acids. Palmitic acid and the stearic acid act as precursors for the 2 most common respective monounsaturated fatty acids, i.e., palmitoleic acid, 16:1, 9, and oleic acid, 18:1, 9. The double bond is introduced by the enzyme fatty acyl CoA desaturase in the smooth endoplasmic reticulum. The process of unsaturation also requires O2, NADH plus H+, microsomal cytochrome B5 and its flavin adenine dinucleotide, FAD, linked reductase. Further desaturation and elongation may create a variety of polyunsaturated fatty acids. 12.6 Biosynthesis of triacylglycerol, triglycerides. Synthesis of triacylglycerol, TAG, takes place mainly in the liver and adipose tissue. It involves the following steps. 1. Synthesis of glycerol 3-phosphate. There are two pathways for glycerol phosphate production. In both liver and adipose tissue, dihydroxyacetone phosphate, DHAP, is reduced by glycerol phosphate dehydrogenase to glycerol 3-phosphate. Dihydroxyacetone phosphate is mainly derived from glycolysis. Dihydroxyacetone phosphate plus NADH plus H plus glycerol 3-phosphate plus NAD plus 2 in the liver but not in adipose tissue. Free glycerol is converted to glycerol phosphate by glycerol kinase. This reaction also takes place in lactating mammary gland, heart, and kidney. Glycerol plus ADP glycerol 3-phosphate plus ADP. 2. Conversion of a free fatty acid to its activated form. Fatty acids are provided by the diet and by biosynthesis in the cytoplasm of adipose tissue or liver. Acetyl-CoA is the precursor for fatty acid synthesis. A fatty acid must be converted to its activated form, attached to CoA, before participating in triacylglycerol synthesis. This reaction is catalyzed by fatty acid acyl-CoA synthetase, thiokinase. RCH2CH2CH2COOH plus CoA plus ADP RCH2CH2CH2CSCoA plus AMP plus PPI. Fatty acid fatty acyl-CoA. 3. Synthesis of triacylglycerol from glycerol phosphate and fatty acyl-CoA. TAG is synthesized by reacting glycerol 3-phosphate with fatty acyl-CoA. The reaction is catalyzed by acyltransferase to first give lysophosphatidic acid in the addition to the first fatty acid carried by fatty acyl-CoA. It is then converted to phosphatidic acid with the addition of second fatty acid. The phosphate group is removed from phosphatidic acid by the phosphatase enzyme and the third fatty acid is added by the action of acyltransferase to give a triacylglycerol. Figure 12.21 Biosynthesis of triacylglycerol, triglyceride. Table 12.1 Plasma levels of triacylglycerols, triglycerides. Values in mg/dl. Increased in desirable 150. Borderline high equals 150 to 199. High equals 200 to 499. Very high equals 500. Hyperlipoproteinemia type I, IIB, 3, 4 and V. Liver diseases. Alcoholism. Nephrotic syndrome. Hypothyroidism. Poorly controlled diabetes mellitus. Pancreatitis. Anorexia nervosa. Decreased in malnutrition. Hyperthyroidism. Brain infarction. COPD. Congenital beta-lipoproteinemia. Exercise questions. For MCQ 6 to 10 see Part 2 and for MCQ 11 to 15 see Part 3 of this chapter. MCQ 1. A 56-year-old man suffering from ischemic heart disease consulted a cardiac OPD to evaluate his condition. Which of the following test would be most suitable to evaluate his condition? Serum AST. Serum cholesterol. Serum LDH. Serum sodium. Serum troponin. MCQ 2. A 64-year-old man suffering from hyperlipidemia consulted a cardiac OPD for a follow-up checkup. He was advised to check his lipoproteins levels. Which of the following lipoproteins is most likely reduced in this patient? Chylomicrons. High-density lipoprotein. Intermediate-density lipoprotein. Low-density lipoprotein. Very low-density lipoprotein. MCQ 3. A 34-year-old man complained of chest tightness and pain on exertion. The pain subsides on rest. History, ECG, and other investigations reflect that he is suffering from angina pectoris. Which of the following metabolic processes is the main source of energy for the affected organ during ischemia in this patient? Fatty acid oxidation, glycogenolysis, glycolysis, HMP shunt, ketogenesis. MCQ 4. A 43-year-old man is brought to ER in an unconscious state. Immediate lab investigations showed blood glucose 280 mg/dl, and serum electrolytes within normal range, but urine was positive for ketone bodies. Which of the following substance is present in the urine of this patient? Acetoacetate, citrate, lactate, pyruvate, malate. MCQ 5. The lab reports of a 56-year-old man suffering from dyslipidemia showed increased levels of VLDL. Further investigations revealed glucose intolerance and hyperinsulinemia. Which of the following hyperlipoproteinemias is most likely present in this patient? Familial hyperalpha-lipoproteinemia, familial hypercholesterolemia type 2A, familial hyperlipoproteinemia type 3, familial hypertriacylglycerolemia type LV. End of Part 1 of 3 of Chapter 12. Thank you.